Hello everyone, welcome back once again to this educational channel called Learn Bio with Janet. I'm teacher Janet and today we'll be discussing types of immunity, subtopic 11.3. This is the second video on types of immunity. And the syllabus involved is the Form 4 Biology KSSM syllabus, Chapter 11. So in this video we'll be discussing in more detail passive immunity, and then we'll compare active immunity with passive immunity. Other than that, we'll look at a few interesting hot questions on this topic at the end of the video. The learning outcomes for this lesson, for this subtopic of 11.3 types of immunity, are as follows. Firstly, we should be able to communicate about the types of immunity, that is passive immunity. Right? And passive immunity is divided into natural passive immunity and artificial passive immunity. Now, active immunity has been discussed in the previous video before this. Okay. Then, other than that, we are going to compare and contrast the passive immunity and active immunity. Firstly, let's recall the definition for immunity. So, immunity is defined as the ability of an organism to resist infection or disease and to defend itself or himself or herself against infection by pathogens. That means that if pathogens were to try to infect the body or invade the body, the organism who has immunity will be able to defend itself and destroy the pathogens or neutralize the toxins so that he will not fall sick from the disease. Okay? Is able to resist the infection and to resist the attack by the pathogens. So immunity depends on the presence of lymphocytes. Okay, this white blood cell with the round nucleus, which can produce specific antibodies during infection. And this is called a specific immune response. So the antibodies that are produced by the lymphocytes help to promote the destruction of the antigens, which are foreign substances that have entered the body, okay, such as pathogens or even toxins. Let us recall the different types of immunity. So immunity can be divided into two main types. Number one, active immunity, and number two, passive immunity. In active immunity, a person's body or his lymphocytes produces his own antibodies when stimulated by antigens or when stimulated by foreign substances that have entered the body. But in passive immunity, the person's body does not produce his own antibodies. He receives the antibodies from an external source, for example, from his mother or from an injection of antiserum. Now, active immunity can be divided into natural active immunity and artificial active immunity. In natural active immunity, a person falls sick when infected, and this will cause the lymphocytes to produce specific antibodies against the specific antigens, like pathogens. Then the antibody level, the level of antibodies will increase until it reaches the level of immunity and is able to destroy all these antigens. So he recovers from the infection and he has acquired natural active immunity after he recovers. And you will have a high level of antibodies against that particular antigen, such as pathogens. Now, in artificial active immunity, an injection is given. Okay, if this artificial immunity, injection is given. So in this case, for artificial active immunity, a vaccine, which are the dead or weakened pathogens, or even the toxins that are modified, is injected into the body to stimulate the body to produce its own antibodies. But the person does not fall sick because the vaccine contains the dead or weakened pathogens. Now, for passive immunity, it is divided into natural passive immunity and artificial passive immunity. Now, for natural passive immunity, the baby receives antibodies from the mother through breastfeeding, or the fetus receives antibodies from the mother again, but this time through the placenta, and it's during pregnancy. For artificial passive immunity, again, when it's artificial immunity, an injection is given. But this time the injection is an injection of antiserum containing specific antibodies. Okay, and this is injected into the body directly to destroy the pathogens or neutralize the toxins that may be found in the body. So for this video, we are going to discuss these two types of immunity, the passive immunity, that is natural passive immunity, and artificial passive immunity. And the video for active immunity has already been done earlier. That is part 1 of types of immunity. Let us discuss passive immunity in more detail. In passive immunity, the person's body does not produce its own antibodies, because the antibodies were obtained from an external source, such as from mother or from injection of antiserum. So the word passive here means that the person's body was passive and did not take part in producing antibodies. Its lymphocytes were not stimulated to produce antibodies. Okay, so the antibodies are from an external source outside the body, and passive immunity does not persist because the foreign antibodies will be broken down in the body of the person. Alright, and it is not replaced unless a second injection is given, or unless the mother continues to breastfeed the child and provide the maternal antibodies to the child. So passive immunity does not persist and can only give immediate and short-term or temporary protection to the person. So there are two types of passive immunity: natural passive immunity and artificial passive immunity. First, let's look at natural passive immunity. Natural passive immunity is acquired by a child when his own mother gives him the antibodies, the maternal antibodies. So there are two ways for a child to acquire natural passive immunity. The first way is when the child is still a fetus in the uterus of the mother. So this immunity is acquired by a fetus when the mother's antibodies diffuse from the placenta here through the blood vessels in the umbilical cord to the baby. Okay, so the antibodies diffuse from the mother's blood into the fetal blood. Alright, and then when the baby is born, the baby can also acquire natural passive immunity. Okay, so natural passive immunity is acquired by a baby through breastfeeding when the antibodies in mother's milk and in colostrum is absorbed by the baby to protect the baby for the first few months after birth. Okay, so the antibodies are in the breast milk. Now, what is colostrum? Colostrum is a form of the mother's milk produced in the first few days from the mammary glands just after the mother has given birth. So it is a yellowish colored fluid, and it is rich in antibodies. So it helps to protect the baby from pathogens by promoting the destruction of pathogens, specific pathogens that have entered the baby's body. So we can say that this is a form of mother's care and love that is natural, because it helps, the antibodies help to defend the baby and protect the baby from attack by pathogens for the first few months. Okay, the specific antibodies from the mother's milk. Now, what is the function of the maternal antibodies to the baby? So the maternal antibodies given to the baby will help the baby acquire natural passive immunity, and it will give temporary immunity to the baby for a few months to protect the baby from disease, because the antibodies can destroy pathogens that try to infect the baby, until the baby's immune system is fully functional. So at birth, the baby's immune system is not yet fully functional or developed. Okay, it needs time to be fully functional. And so in the meantime, the maternal antibodies can provide protection for the baby. So this is a form of natural form of mother's love, in the sense that the mother defends the baby from disease. All right, and we have to thank our mothers for this. Okay, number 3. Why is natural passive immunity only temporary? Okay, common question. This is because the mother's antibodies are foreign antibodies to the baby. So when they are in the body of the baby, it will be broken down. The antibodies will be broken down and they are not replaced. All right, because the baby's lymphocytes were not stimulated by any antigens or pathogens to produce antibodies, specific antibodies. All these antibodies came from the mother. All right, so there are no memory cells produced that have a memory of the antigens and that can produce the specific antibodies for the baby. Okay, so the mother's foreign antibodies, the mother's antibodies which are foreign to the baby, will be broken down and are not replaced. So the baby will then lose the natural passive immunity after some time, should the breastfeeding stop. Okay, then the natural passive immunity will be lost after a few months. Then the baby's immune system will have to start to build its own, start to build immunity for the baby. Okay, so the baby will build immunity or through the injection of vaccines, and also through falling sick and then recovering. For example, falling sick from a cough or cold, from flu, and then recovering from it. And that way he will have build his own active immunity. Let us look at the graph for natural passive immunity. So imagine the mother starts to breastfeed the child. All right, so when the child is born, the mother starts to breastfeed the child. So the antibodies from the mother will be given to the baby or fetus, and the level of antibodies will increase quite fast. All right, because the antibodies are ready-made from the mother, and the child does not need to produce his own antibodies. All right, which will be more gradual, which will increase the concentration of antibodies more gradually. Right, so the level of antibodies will increase until it reaches the level of immunity and beyond. But if the mother should stop breastfeeding the child after a few months, the concentration of antibodies will decrease rapidly as the foreign antibodies are broken down and are not replaced. All right, so the graph is quite steep, goes up quite steeply and then comes down very fast. All right, let's compare this with the graph for natural active immunity. Okay, so the white curve is the graph for natural passive immunity, and the blue curve is for natural active immunity. So in natural active immunity, the baby may fall sick from some bacterial infection or viral infection, and his lymphocytes will start to produce antibodies, will be stimulated to produce antibodies in order to destroy the pathogens. So the increase in the concentration of antibodies will be gradual as the lymphocytes produce more and more antibodies until it reaches the level of immunity and becomes immune. All right. After that, the baby recovers from the sickness, and then the concentration of antibodies can decrease again if there is no more stimulation or no more infection by the same pathogen. Okay, but it will decrease slowly as the lymphocytes of the baby are the ones that produce the antibodies. Okay, so the curve for natural active immunity is the curve or the gradient of the curve is gentler than the one for natural passive immunity. The second type of passive immunity is called artificial passive immunity. So when it is artificial immunity, it involves an injection of a substance. In this case, for artificial passive immunity, it is acquired through an injection of antiserum, okay, or we can say it's antibodies in a serum. So an antiserum is defined as a serum that contains specific antibodies to fight specific antigens. And a serum is just blood plasma without fibrinogen, which is a substance that's involved in blood clotting. Okay, so the antiserum actually contains antibodies, okay, to directly promote the destruction of pathogens or to neutralize toxins that are in the body of the patient. So this antiserum can be obtained from animals, as we will discuss later, and or also even from people who have successfully recovered from a disease. All right, they will have the antibodies for that particular disease. Okay, for example, it has been reported that to treat the COVID-19 disease, some areas or hospitals use the antibodies from the patients that have successfully recovered to help those who were sick with the COVID-19 disease. All right. So the antibodies will directly promote the destruction of the pathogens or neutralize the toxins in the patients who are sick. Now, it is usually used, this antiserum is usually used to destroy pathogens that have already infected a person. So it is used, it is injected into sick people or those that are at risk of being infected with the disease, okay, or suspected of being infected with the disease. So it gives quick and immediate protection, but only for a short period of time. And we say that this type of immunity is temporary immunity or short-term immunity. What is the reason that it is only temporary? Okay, it's the same reason as for natural passive immunity. Okay, it's because the antibodies are foreign. It's from an external source, not from the body of the patient itself, himself or herself. So these foreign antibodies from external sources will break down in the body of the person, and they are not replaced, as the person's own lymphocytes were not stimulated to produce these antibodies. Okay, and there are no memory cells produced, no memory lymphocytes produced that have any memory of the antigens. Let us discuss a few interesting examples of antiserums that have been developed so that a person can acquire artificial passive immunity through the injections of these antiserums, should he need them. So examples of antiserums are antitetanus serum, anti-rabies serum, and antiserums for the different toxins of poisonous snakes that may bite people. Now, these antiserums contain the ready-made antibodies to either destroy the specific pathogens of a disease, or sometimes it's to neutralize the dangerous toxins, okay, produced by the pathogens of that disease. So 4 examples are given here for the antiserums that have been developed. So for rabies, now rabies is sometimes called the mad dog disease or penyakit anjing gila. So it is a viral disease, that means caused by a virus, transmitted through bites of wild animals, and it attacks the nervous system of a person. So it can be fatal. So a person who has got has been bitten by a wild animal or even a dog is advised to seek a doctor to get injections of the antiserum for rabies, so that the antibodies in the antiserum will immediately work to destroy the virus for rabies in case it has entered the body. All right, so that the person will not suffer the bad effects of this disease, okay, and it may save his life. Now, botulism is caused by a toxin from a bacteria that attacks nerves, and it is transmitted through food that is not well preserved. For example, food that is from a can or tin that may not be sealed very well. So again, now in this case, it's the toxin that is dangerous. Okay, it's produced by the bacteria. So if a person has botulism, we can be given the antiserum for botulism to so that the antibody in the antiserum, which is an antitoxin, can neutralize the toxin, okay, of the bacteria, okay, to prevent it from attacking the nerves. Now, tetanus or lockjaw disease is caused by a bacterium in the soil that produces a dangerous toxin also, and is transmitted through cuts caused by rusty objects. For example, when a person steps on a rusty nail or gets cut by a rusty knife. So there's a possibility that the bacterium may have been transmitted to him. Okay, so he's advised to get an injection or injections of antiserum for tetanus, and again this antiserum will contain the antibody or that's called the antitoxin for tetanus, to neutralize the toxin, okay, of this tetanus, and to help the person recover from the infection by the pathogen in case he has been infected. Now, snake bites contain toxins or venom. Okay, venom means the toxins from the poisonous snakes. So if a person gets bitten by a snake, again he has to be rushed to hospital to be injected with an antiserum which contains the specific antibody or antitoxin to neutralize the toxin from that specific snake. Let us discuss something interesting here. How is an anti-tetanus serum prepared? Now, first of all, a tetanus toxoid suspension is prepared. So a toxoid is a toxin that has been modified and is no longer toxic, but it can be used as a vaccine. So this toxoid is injected into a horse, and it acts like a vaccine in that it will stimulate the horse's lymphocytes to produce antibodies to neutralize the tetanus toxins. So after the horse has produced the antibodies, the horse's blood is drawn out and the plasma is separated from the blood cells. Okay, so where are the antibodies? They are contained in the plasma or the serum from the horse. So the anti-tetanus antibodies from the horse's serum is concentrated and then purified. So the solution or the serum will contain a high concentration of the antibodies that can neutralize the toxins of tetanus. Next, it can be used by a doctor, and it is injected into a person who has a high risk of tetanus, or a person who has already got a deep cut or a cut contaminated with mud or soil, because the bacterium is found in the soil, right? So a person who has been cut by a rusty object also, okay, will be injected with this anti-tetanus serum. So the antibodies will then be able to neutralize any toxins produced by the tetanus bacterium. So the person is protected from tetanus infection. Let's have a look at this graph for artificial passive immunity obtained through injection of anti-serum. So the graph is very different from the one for injection of vaccine, for artificial active immunity. This graph goes straight up because when the doctor injects the sick person with the injection of antiserum, in the first injection, he will make sure that the antibody level is very high and beyond the level of immunity in order to destroy the pathogens effectively, the pathogens in the body. So after that, the level of antibody will decrease very fast because these are foreign antibodies that are broken down by the person's body. Next, the doctor may give a second injection to boost up the level of antibodies beyond the level of immunity again. So it's called the booster dose of antiserum. And this is again to destroy any remaining pathogens that may be able to still cause harm to the patient. Okay, so the person will have temporary immunity from the disease. And the level of antibodies decreases very fast compared to artificial active immunity. So the graph is something like shark's fin under water like that. So remember the shape. This is for artificial passive immunity. The shark's fin shape, right? For artificial active immunity, the graph is like two peaks, one lower and one higher one. Okay. Let us look at this Soalan Ramalan or forecast question for SPM. Now, based on graph X and Y below, compare and contrast the two types of immunity that's acquired. So read the question a few times. Based on graphs X here and Y here, compare and contrast, meaning give the similarities and the differences between the two types of immunity that's acquired. So what type of immunity is acquired for in graph X and in graph Y? We have to identify that. Okay, so read the question carefully and look at the graph. It shows the concentration of antibodies against time. And then there's the level of immunity here. All right. So first of all, let's identify what type of immunity is involved in each graph. If you look at graph X, it has the shape like two mountain peaks, one lower, one higher. So in graph X, the person was injected acquires artificial active immunity, right? And graph Y looks like shark's fin underwater, with this part underwater. So the person was injected for graph Y, the person was injected acquires artificial passive immunity. All right. Next, you can state the similarities and the differences. So each similarity is 1 mark, each difference is 1 mark. So have a combination of both similarities and differences. Now, compare and contrast questions are popular in the SPM. So make sure you know all these types of compare and contrast questions for different topics. Okay, similarities. So in both cases, for graph X and Y, it involves an injection. They involve an injection of a substance, and the aim is to protect the body from infectious disease. Okay. Secondly, both involve the use of antibodies to destroy antigens, or there is an interaction between antibodies and antigens, where antibodies destroy or promote the destruction of antigens in the body. In both cases, antibodies are involved. Thirdly, in both cases, the level of antibodies must exceed this level of immunity for the person to acquire immunity. If the level of antibodies is below the level of immunity, the person is not immune yet. And fourthly, in both cases, there are booster doses given to increase the level of antibodies so that exceeds the level of immunity. Let's go on to the differences. Now, try to fill in the blanks here, all right, as I read. So what are the differences between graph X and Y? Now, the graph for graph X shows what type of immunity is acquired. Artificial, active immunity, right? For graph Y, the graph shows how artificial, passive immunity is acquired, right? Now, graph X involves injection of what substance? What type of substance? Which are dead or weakened pathogens. Vaccine, a vaccine. For graph Y, involves an injection of what type of substance? A serum that contains specific antibodies. Antiserum. Now, don't just say that it involves injection of a serum. A serum is just blood plasma without fibrinogen. It doesn't mean that it has antibodies, specific antibodies. So must say antiserum, which means antibody in the serum. Now the person acquires immunity only after the second injection or booster dose. So here we have these words, either immediately or gradually. Okay, so this one is what? Acquires immunity gradually, only after the second injection. So during after the first injection, he didn't acquire immunity. He only acquired immunity after the second injection. Here, after the second injection, he acquires immunity here, when the level of antibodies reaches the level of immunity. But for graph Y, the person acquires immunity immediately after the first injection. So the doctor will ensure that the antibodies that are injected into the person is at high concentration. So he will gain immunity immediately in order to destroy any pathogens that may have entered his body. Okay, for the second graph. Because here we assume that the person may have been infected with the pathogens. Whereas for graph X, a healthy person is injected with the vaccine. He's not sick, or there is probably a low risk of him having the pathogens. There's no risk of him having the pathogens. He's a healthy person. Now, number 4, antibodies are produced by the person's own what? For graph X. What cells? Lymphocytes. But for graph Y, antibodies are obtained from where? From antiserum. From an antiserum, which is ready-made antibodies. Okay, so it's from an external source. Now let's discuss more differences. Number 4, the immunity lasts for a longer or shorter period of time for graph X. Longer period of time. You can see it here at the end of the graph. The slope is quite gentle, the gradient is quite gentle, meaning that the level of antibodies decreases very slowly, and when it has the level is below the red line, then the person is no longer immune. Okay, but it takes a while for the level of antibodies to decrease below the level of immunity. But for graph Y, the immunity is temporary or permanent? Temporary. And is it long-term or short-term? Short-term. It does not persist compared to the immunity shown by graph X. Okay, I can see here that the curve, the gradient of the curve is steeper, and this shows that the level of antibodies decreases very fast until it's below the level of immunity. Then the person will be no longer immune to the disease. So the injection is given to a healthy person for graph X, and that is to what the infection? Is it to prevent the infection or to treat the infection? Okay, it's to prevent infection because the person has not yet been infected with a particular pathogen. So the vaccine prepares him, because the vaccine will stimulate the lymphocytes to produce the antibodies for that particular pathogen. Okay, so that he's ready to prevent infection in the future, should the real pathogen infect him. Now, for graph Y, the injection is given to a sick person as a what? Or for immediate protection? As a treatment. Okay, assuming that the person is already sick. Okay, now let's look at number 6. The second injection of vaccine or booster dose is given to increase the level of antibodies here beyond the level of immunity, so that the person becomes immune and is protected from the disease. Okay, once the level of antibodies exceeds the level of immunity, the person has become immune and is protected from the disease. It is not to destroy any remaining pathogens, because the person has not experienced any infection by pathogens yet. It's just to make him immune. Now, the second injection for graph Y is the injection of the antiserum. The second injection of antiserum, or the booster dose of antiserum, is given to increase the level of antibodies too, to exceed the level of immunity. But the aim is to help the body destroy all remaining pathogens if the patient is still infected. Okay, let's say the patient was infected here and the doctor has already given him the first dose of antiserum or antibodies, but it may not have destroyed all the pathogens. So after a while, the doctor will give him another injection of antiserum to boost his level of antibodies beyond the level of immunity in order to destroy all the remaining pathogens in the body. Okay. Okay, last two differences here. Number 7, memory cells or memory lymphocytes are produced for graph X, which will produce a more rapid immune response during future infections. So because the vaccine injected has stimulated the lymphocytes to produce antibodies, specific antibodies against that specific pathogen or antigen, thus some of the lymphocytes will remain as memory cells, which will be there through the years, so that they can produce a more rapid immune response by producing large amounts of antibodies very quickly should there be future infections by the same pathogen. Okay? But in graph Y, the memory cells are not produced for this type of immunity. Alright? Because the antibodies came from an external source and the lymphocytes of the person were not stimulated to produce the antibodies themselves. So there are no memory lymphocytes produced. Number 8, we can also give examples, alright, as part of the differences. So examples of vaccines are those for rubella, hepatitis B, measles, tuberculosis, polio, mellitus, and others. For graph Y, examples of antiserum are those for tetanus, rabies, and snake bites. Right, now a tip from A+ students. So some A+ students write down essay answers that are good as practice in an exercise book to remember the points. And this will train you to write essays more confidently and more easily during the real exams, okay, and during SPM. Here are some interesting HOTS questions that are similar to the ones that come out in exams, right? So let's read through this and try the questions. Number 1, explain the difference between an antibody and an antibiotic. 3 marks. Secondly, state 2 ways in which the COVID-19 virus is transmitted. What are 3 effects of the virus on the health of a person? And state 3 steps to take to prevent the spread of COVID-19. So it's important to know about COVID-19 as it is causing a pandemic now. And pandemic means a disease that is spread throughout the whole world. So we need to know all the facts about it. Number 3, why is it important to develop a vaccine for the COVID-19 virus? So what's the importance of a vaccine for this new disease? Number 4, through what process can a virus render a vaccine against it ineffective? So how does the virus fight back? Should a vaccine be developed for a virus like a COVID-19 virus? Okay, 3 marks. And number 5, a boy is bitten by a fierce stray dog. His parents are worried that he may be infected. What should they do? Okay. Number 6, state 3 diseases that a baby in Malaysia is vaccinated and immunized against in the first year of life. So we need to know a bit about the immunization program for babies and for children. Predict what happens if the baby was not vaccinated. Okay, prediction questions also important. So try these questions and answers will be posted in the description box in about 1 to 2 days time. Alright, so you look through the questions first once the video is posted and try the questions yourself first. Right, the answers will be posted in the description box in 1 or 2 days time. Just keep a lookout for it. Okay, so that's all for this lesson. In the next lesson, we will discuss subtopic 11.4, health issues related to human immunity. And we'll discuss some diseases which are related to the immune system of humans. So thanks for watching. Stay tuned for the next video and please share, like, and subscribe. Goodbye for now.